Thanks for joining us, Olga. I was wondering if you could tell us about the research project, a little bit about the timeline and what you hope it will achieve, the patient benefits that will come of it.
Thank you for actually arranging this kind of nice, virtual talks. I think it's important to give you an update of where we are in my group and how we managed to use the funds that we were allocated from Make Seconds Count. So we have two projects which are currently running. I'm sure you know that there were significant difficulties because we were not allowed to work during the COVID lockdown. So we've only started at some point in September going back to the labs. So we have two very promising projects, and there are a senior postdoc fellow, which is funded by Make Seconds Count for a 3-year job, and 2 PhD students, along with a research associate. There are two projects, and I'm going to refer only to the projects on the metastatic setting, because obviously we have other projects running on the early setting of breast cancer. I'll give you an overview of the projects and what we're trying to achieve. So the breast cancer has lots of different profiles, which each of them requiring a completely different management. I'm sure that all of us and all of you have heard of triple negative breast cancer, ER positive breast cancer, HER2 positive breast cancer. They all differ. They all have a completely different biology, which means that we need to identify what might be the best treatment once we understand the biology of each of these cancers. Not all tumors will behave in the same way, and some might be very sensitive to treatments, some others might be very resistant to treatments. And this behavior depends on a set of genes that drive the cancer cells. So we just need to identify which are these genes and how they behave. So we believe that the most unstable these genes are, the more aggressive the cancer cells might be. So based on that, we have one project which is running on the ER positive, so the luminal breast cancer. We have managed for several years now to recruit lots of patients in our studies. So we have a very large cohort of tissue samples and of blood samples, longitudinal samples from patients throughout their journey with breast cancer, which means we have the chance to look at the tissue blocks or the blood from when the patients had early breast cancer. And then we have matching samples, from the same patients, from the recurrent metastatic sites. This gives us the opportunity to look in depth what might be the differences, first of all, between the early diagnosis and the metastatic, gives us the opportunity to see how unstable these genes are, and perhaps explore and identify what might be the best treatment and save the patients from receiving treatments that obviously they don't come without any toxicities, but identifying the treatments that suit each of these patients rather than giving a treatment that is for ER positive breast cancer. So we are aiming to identify on this ER positive cohort of patients, so the patients that they have sensitive disease to hormonal treatments, whether they will respond well to the hormonal treatments, plus these new fantastic agents, which are called CDK4/6 inhibitors, or save them from the unnecessary toxicity if we identify early that these patients actually need chemotherapy. So that's one of the projects, that is focusing on hormone-sensitive breast cancer samples and obviously breast cancer patients. The other one now is about the triple negative breast cancer. The triple negative breast cancer is perhaps the most aggressive subtype of breast cancer. The patients with metastatic breast cancer require chemotherapy because at the moment we don't have any specific targeted agents which have been approved by NICE, SMC, FDA or EMA. So these breast cancers, they tend to be recognized by, or they tend to show significant chemosensitivity, which means they respond well to chemotherapy. However, within a short time, something happens and they stop, or they become extremely resistant, and then we need to change to a different treatment again and again. These breast cancers are characterized by a significant tumor heterogeneity. Let me explain you what that means. It means that within the tumor block, we might have different types of cells. Some express a set of genes in one area, let's say, of the tumor, which might actually respond very well to the chemotherapy. Some others, they might not, or they might become resistant. So we want to understand whether this kind of resistance to chemotherapy might be either due to this intratumor heterogeneity, as we call it, so the different clones of genes, different kind of information within the same group, or it might be the result of changes that happen as the result of the chemotherapy drugs, immunotherapy, and all these kind of cytotoxic agents that we might offer to these patients. So we're trying to find out whether these were the resistance might be due to pre-existing changes on the tumor environment or due to the emerging of new genomic aberrations, new genomic changes, new changes as a result of the cytotoxic treatment.
Yeah, it sounds very much.
And so,
it sounds very much like pioneering work. Has anything been done like this before?
No, and I'll tell you what is the significant advantage that, let's say, we have this group. So because of all this work, the last perhaps 7-8 years since I moved to Edinburgh from London, where I was trained, I set up this, I would say, really difficult pathway, trying to collect samples and clinical information from the patients. We have to acknowledge that we don't tend to require or to request biopsies from metastatic sites. And this is because most of the biopsies or some biopsies might be extremely risky for the patients. So very careful consideration is given when someone is requested to have a biopsy from the recurrent metastatic site. But we do recognize that some of the cancers might actually change in the matter of the time from the initial diagnosis till the metastasis. So they might actually change their profile. And that's why biopsying the recurrent site is extremely helpful when it comes to assess and study the biology of the cancer. So we managed to actually recruit a significant number of patients and to have this kind of longitudinal samples in our both tissue and database, because it's always important not just to have the tissue, Colin, I'm sure you understand, because the tissue without the clinical information is nothing. It might be important, yes, but when you're trying to bring the knowledge from the bench to the bedside, which means to the actual clinic, you need to have access to the whole clinical information about each of these patients. So we have a significant number of samples, which makes our studies very powered. And I'll tell you why powered means it's extremely significant. First of all, because it will probably allow us to get a significant funding from some grants that we have already applied, because this is one of the questions always in the grant proposals: how powered is your study? If we had, let's say, 10, 20, 30 patients, it wouldn't have been enough, because yes, we might come up with something really important, but again, 30 patients is not enough. We're talking about lots of hundreds of patients and lots of longitudinal, so sequential samples from these patients throughout their journey from the early cancer to the metastatic site. So that's definitely a very strong card that we have in our sleeve. So we have already applied and we managed to get significant, a very good funding, I would say, a 6-digit funding so as to run one of these projects. And we are trying now to finish up and submit another application. But we have managed also to establish a very good collaboration with Karolinska, which I'm sure you know that is a very well-reputed university and institute for breast cancer. So not only we are working with our samples and we're trying to explore our ideas, but we're trying to collaborate with really big centers, translational researchers in breast cancer, which I think will make our case even stronger and our results even more significant, because we are combining, the power of two big centers and obviously the number of the samples.
Yeah, yeah. I mean, you've mentioned funding there, and these studies are expensive, and a lot of people who will be watching this interview have supported Make Seconds Count. So can you emphasise the importance of the role that charities such as Make Seconds Count play in establishing this type of research project?
Absolutely. First of all, the Make Seconds Count gave us the opportunity, as I said at the very beginning, to recruit a senior postdoc fellow. The money that we received, which was, lots of money, and there was a significant funding, managed actually to secure a 3-year post. And I can tell you that the senior postdoc fellows are, extremely well paid, I would say, especially on the grade that we aimed to recruit someone. Mm-hmm. So we've used all the money, and we actually had to add even more so as to manage to offer that position to the person that actually got the job, because we felt very strongly that that's the right person. Most of the grant applications, as they are standing now, because the COVID has affected significantly the grant proposals, as you know, CRUK Breast Cancer now actually withdraw lots of the applications or calls that they had available for this year. So there are some grant calls, but these are mainly focusing on funding the consumables or funding the techniques of the experiments rather than funding the people. So I think by making the decision to actually use the money to fund the position rather than using the money for consumables, puts us in a strong position of being able to say, we would like this money to fund this proposal. We do have the staff to do it. We have the material, the patients, and all the tissue to actually do it. So we just need some help in terms of doing all these very expensive experiments and fund the consumables. So definitely without Make Seconds Count and without all this fantastic effort, because I've been there since day one of the Make Seconds Count, or even before the idea and before the setup of the Make Seconds Count, and I could see how much hard work is behind the scenes, and sometimes people might not even see it, but there is so much passion behind every single person that I've met in this charity about making that bigger, successful, and we've seen so far that you have achieved loads, not just,
Yeah, and this project. This project's very much a team effort, and that includes patients, obviously. Could you give me an idea of the scale of the project in terms of your team and how many people are involved directly and indirectly?
Are you talking about the people that they are working in the projects or the patients that they have been recruited to the
both
studies?
I think both. If you could split it into two and give us an idea of the numbers.
Yes. So we have 4 people dealing with the 2 projects on the metastatic setting, and we're going to have a 5th one pretty soon. So we're talking about 1 senior postdoc fellow, which has been funded, as I said, from Make Seconds Count, 2 new PhD students, a research associate, and we're having an MSc student joining us soon. In terms of the patients now, and I think it's a good idea that you're asking me, because I think we need to inform the patients that, persuade them that participation to this kind of translational studies is equally important to participation to clinical trials. And let me tell you what might be the difference. So the clinical trials investigate a new experimental drug. And all the patients, especially the metastatic ones, are extremely keen to participate to a clinical trial, especially when they have exhausted the standard options. Translational studies now are something different. Translational studies rely on patients being keen to donate a blood sample, a biopsy when possible, for us to run our experiments and actually try to study the questions that each of the projects addresses. So it's really important for the patients to be open and think about, yes, the translational study might not have an immediate impact on me, but it might help me down the line and might actually identify something which might be helpful. I have to say that there are some patients that they had this kind, I'm going to talk about patients that they were on a different study. One of my studies, which I have published last year, this was on the neoadjuvant setting, so on the very early setting when patients receive chemotherapy so as to shrink the tumor. So we managed to identify, when these patients came back with metastases, because we had these samples, we managed to identify specific gene mutations, and we managed to actually allocate these patients to clinical trials that were targeting these specific mutations that we identified through our experiments in the lab. But because I don't want this to be misunderstood, after we identify, because obviously whatever you identify in the lab, Colin, you cannot take it straight away to the clinic, because this is research, okay? But this gives us an idea of whether we need to investigate further with perhaps testing, genomic testing, that they have been approved and authorized for that use, and then have a very solid interpretation or identification of the mutations and how these patients might benefit as a result of this. We call it next generation sequencing. So not that the lab research will actually lead to changes of the management, but it might give us an idea of the next step to be taken so as to offer the patient the best possible treatment. And this is called personalized medicine. And that will be the future in cancer, not just in breast cancer. The identification of all these genes that they actually drive the cancers.
I mean, you sound very excited by the project, as we are. At what stage will you be able to give us an update on how it's progressing? I know it's against the backdrop of COVID, but what are your hopes if you've readjusted them at the moment?
So, as I said, these are two different projects running, which require significant input from bioinformatics. So, because yes, we have the tissue, we do all the tests, we need someone to sit down, an expert, which is called bioinformatician, to sit down and analyze all these large data set, which is, it's not within our expertise, but we have a very strong collaboration within IGMM, and without them, we wouldn't be able to interpret. So we wouldn't be able to actually identify these changes. So it's a very exciting project, but nothing comes up within, a month or 2 months, 3 months. So I would say that in 6 months' time, we will be able perhaps to say where we are in terms of how much we have achieved in terms of identifying the patients, setting up all the experiments, decide exactly what needs to be done, do all the extractions, and decide on what is the best methodology of checking each of these aims of the project, because no project has just one objective, they all have multiple objectives. And because we're talking about hundreds of patients, let's not ignore that. Very careful, management of the funding, which obviously will depend on, successful applications. So there are so many parameters, but we hope to be in a good, level, perhaps in 6 months' time. And the aim is to plan 6 months ahead what we're going to do. But again, you just need to be alert, and some things might not work, and we might have to change a few things or a methodology. But we feel very, I would say, very optimistic. We are very optimistic that things will go very well, because we do have the expertise on all these kind of techniques and experiments. It's not that we're trying to set up something completely new from the beginning.
Okay, well, we wish you and the team every success, and we look forward to hearing how it's progressing. And it's going to be of fantastic benefit to patients and supporters of Make Seconds Count. So thank you, Olga.
No, thank you, and thanks everyone supporting Make Seconds Count, Colin. Thank you.